In section 3, we are looking at how DNA replicates. So replication is going to be the copying of genetic information. So a single strand of DNA serves as a template for a new strand. Rules of base pairing, direct replication. So as it's being opened up and copied, if there's an A, it's going to pair with a T. Simple as that. So DNA is replicated during the S phase, or the S stage of the cell cycle. Each body cell gets a complete set of identical DNA. So that S, again, stood for synthesis. That's going to be where it's copying the DNA. Once it's done that, it needs to go through cell division. So proteins carry out the instructions, carry out the process of replication. So DNA serves as a template. There are enzymes and other proteins that do the actual work of replication. So you're going to have an enzyme that's going to unzip the double helix. Free-floating nucleotides form hydrogen bonds with the template strand. So you see it's going to unzip, it'll move both directions when it unzips, and that's both DNA in a eukaryote and a prokaryote. The DNA polymerase enzyme is going to bond the nucleotides together to form the double helix. Polymerase enzymes form covalent bonds between nucleotides in the new strand. So you can see there in that picture that you've got the DNA polymerase, it's moving along and it's attaching those new nucleotides together, and it's making an old strand now paired with a new one. So two new molecules of DNA are formed, each with an original strand and a newly formed strand. DNA replication is semi-conservative. So as you see in that picture, you've got an old strand and a new strand, and so you aren't having to build all from new. That's what the semi-conservative means. Replication is fast and accurate. DNA replication starts at many points in eukaryotic chromosomes. In prokaryotes, it's going to start at 1 and go around and meet, because the prokaryotes, it's going to be a circular ring. In eukaryotes, again, it's going to start at many points. So for example, there, if we look at A, it's then going to proceed to the B and then C, and it's going to keep going until you have all of it copied. As this is happening, DNA polymerase is going to be sort of looking for and finding and correcting any errors that may have happened during the replication process. So those are the notes on replication.